Let's review what we already know. DNA is a polymer composed of nucleotides, and each nucleotide contains a phosphate group, a 5-carbon sugar. It's either deoxyribose in DNA or ribose in RNA, and a nitrogenous base. And these nitrogenous bases that are found in both DNA and RNA are adenine, guanine, and cytosine. Thymine is found only in DNA, and uracil is found only in RNA, but we're not talking about RNA, so we're going to ignore uracil right now. Understanding DNA directionality is extremely important. The carbon atoms in our 5-carbon sugar are numbered 1 prime. You'll also see it written as 1 prime, 2 prime, 3 prime, and so forth. So to do this numbering system, we need to start by finding our oxygen atom here. We're going to go clockwise. So this is our 1 prime carbon, 2 prime, 3 prime, 4 prime, and 5 prime. This numbering convention allows us to have clear and accurate communication when discussing the form and function of nucleic acids, just the same way as following traffic rules keeps us safe on the road. Because nucleotides connect in a specific way, DNA strands have a directionality. One end is called the 5 prime, and the other end is called the 3 prime. This directionality becomes critical when we discuss DNA replication. The 5 prime end of our new nucleotide here is going to attach to the 3 prime end of the previous nucleotide. Just like Legos, the linking together of nucleotides only works when both pieces are oriented in the correct way. Erwin Chargaff discovered an important pattern in DNA sometime in the mid-40s or early 50s. The amount of adenine is always equal to the amount of thymine. He also noted that the amount of cytosine is always equal to the amount of guanine, but the amount of adenine and thymine does not always equal the same amount as cytosine and guanine. The ratios of A's and T's versus C's and G's varies from species to species. These observations became one of the key clues in how to solve the DNA's structure. Hydrogen bonds are what holds the A's and the T's and the G's and the C's together. G's and C's form 3 hydrogen bonds. A's and T's only make 2 hydrogen bonds. Remember that hydrogen bonds are individually weak, but collectively strong. The 2 DNA strands, being held together in the middle by those hydrogen bonds between the base pairs, run in the opposite directions as each other. On this side, the 3 prime end is here, the 5 prime end is here. On the other strands, pointing in the opposite direction. This is just like I-17 that I drive on every day when I go to work. I need to get on the northbound lanes to drive to work. Then I take the southbound lanes when I head back home. The northbound lanes and the southbound lanes run alongside each other, meaning that they're parallel, but they head in opposite directions, meaning that they're anti-parallel. And so too are the 2 strands of the DNA molecule.